Welcome back. So today we are going to discuss about bacterial transformation. Okay, so in your molecular biology syllabus, you have bacterial transformation, transduction, and conjugation, of which this is the video where we will just focus on transformation. Now we know that bacteria, they divide by binary fission, and the genetic material is also copied during cell division, and then it is passed to the offsprings. So this is known as vertical transmission. Okay, so the genetic material is transferred to offspring via cell division. Okay, so equal copies or same identical copies are made and they are passed on to the offsprings. The mutations are transferred from one bacteria to another through horizontal transmission. Okay, so now this is not found in each and every bacterial cell. So mutations, they are random and the frequency can differ. So the transmission of mutations from one bacteria to another is known as horizontal transmission. Okay, so there will be slight changes into the genetic materials. So generally there are three different methods or three different types of horizontal transmission for the transfer of genetic information, that is transformation, transduction, and conjugation. So now about transformation. So transformation is the process of DNA uptake by bacteria from the surrounding environment, and usually an expression of a newly acquired trait is seen in a recipient bacteria. Okay, so what happens from the surrounding environment? Free DNA, if they are present in the environment, then a bacterial cell can take it up and it can, or the DNA when gets expressed, the bacteria can acquire a new trait. Okay, so generally best example for this I will say is antibiotic resistance. Okay, you can test it in your lab also by performing bacterial transformation experiment. The cells that have the ability to uptake DNA, they are known as competent cells. Okay, so this process was first reported in Streptococcus pneumoniae by Griffith, and competent cells are one which are or which have their cell membrane permeability altered. Okay, so competent cells, they doesn't require a living donor and only requires free DNA which is available in the environment. The recipient that successfully propagates the new DNA is known as transformants. Okay, so when the DNA is expressed, you can test your bacterial transformation is successful or not by having some or doing some performing some experiments. And when those kind of bacterial cells, they grow into a colony, you can term them as transformants, as they have acquired the new DNA and it has been propagated. Transformation is adopted as the most common method of gene transfer, as it is best way for transfer of artificially acquired or altered DNA into recipient cells. Okay, so this is a best adopted method to artificially alter DNA into a particular recipient cell and test the piece of DNA that is transferred. Okay, the process of transformation can transfer DNA regions of 1 to 10 of kilobases. Okay, now transformation is based on natural ability of bacteria to release DNA, which is taken up by another competent cells. The success of transformation depends on the competence of the host cells. If your cells are not competent, you could not alter your cells, then there is no success in your transformation experiment. The competence is the ability of cell to incorporate naked DNA which is present into the surrounding in the process of transformation. Now organisms that are naturally transformable, spontaneously they release their DNA in the late stationary phase via autolysis. Now some bacteria, including E. coli, can be artificially treated in laboratory to increase their transformability, means making them competent by using some chemicals such as calcium or by applying a strong electric field, that is a technique known as electroporation, or by using a heat shock method. Now electroporation or heat shock, it increases the competence by increasing the permeability of the cell wall, okay, which allows entry of the donor DNA into the cell. And once the DNA has entered the bacterial cell, it can get incorporated into, say, chromosomal DNA, or if it is plasmid, then it can get expressed by itself. Now similarly, transformants can be selected if the transformed DNA contains a selectable marker such as antimicrobial resistance, or if the DNA encodes for utilization of a growth factor such as amino acid. So you should know the sequence of DNA, and if you know what kind of selectable marker is present on your DNA piece of DNA, then you can test for your transformants. In most of the naturally competent bacteria, the free DNA, it binds to the bacteria, and then DNA is integrated into the chromosomal DNA. Okay, this is in case of naturally competent bacteria. Now sometimes a free bacteria is inserted into plasmid as well, which is capable of replicating autonomously. That means it is not dependent on chromosomal DNA, it has its own replicating factors, so it can replicate autonomously, and thus the insert does not have to be integrated into the chromosome. Now this depends if your cell has plasmids already. If not, then yes, your DNA can get incorporated or integrated into chromosome. Now plasmids, they encode some enzymes and antibiotic resistant markers which are later expressed in transformants after transformation. So in this process of transformation, the donor DNA is first inserted into plasmid, and the plasmid containing donor DNA is then inserted into competent host bacteria. Now this is the another method where you incorporate the piece of DNA into plasmid and then insert that whole plasmid into your competent host bacteria. Okay. After the transformation is completed, bacteria containing the plasmid can be detected either by using growth media supplemented by a particular antibiotic, as plasmids, they generally have a selectable marker for antibiotic resistance. Okay, so this was about the process. Now about making your bacterial cells competent or bacterial competence. Not all bacteria are capable of taking up DNA from the surrounding environment. So such bacteria, they are treated artificially. Okay, that is, we alter their cell wall permeability. This is achieved by using some chemicals or electric pulse. In case of chemicals, the cells are chilled and made permeable in the presence of calcium chloride. They are then incubated with the DNA and provided with a heat shock treatment that causes the DNA to enter the cells. Electroporation is another technique where bacterial cells are subjected to electrical pulse or pulses to make them permeable and cause the DNA to enter into the cells. Now there are 4 steps in transformation. First, you develop competence. Second, binding of DNA to the cell surface. Third, process of uptake of free DNA. And fourth, integration of DNA into chromosome by recombination. These are the 4 most important steps of the process of transformation. Now the artificial development of competence can be achieved either by electroporation or through heat shock treatment. The choice depends on the transformation efficiency which is required. It depends on your experimental goal or available resources. For heat shock, the DNA and cell mixture is kept on ice and then it is exposed to 42 degrees Celsius, say water bath. After electroporation, the mixture is transferred to electroporator. Sorry, for electroporation, the mixture is transferred to electroporator and then it is exposed to a brief pulse of high voltage of electric field. This is how electroporation is done, and this is how heat shock is given. The double-stranded DNA release from lysed cells, it binds non-covalently to the cell surface receptors. Now there is no DNA sequence which is specific for no DNA sequence specific recognition is there on the cell surface receptors. Thus, these organisms can potentially incorporate DNA from outside their species as well. That means from any other species, a DNA sequence can be taken up by the cell. The bound double-stranded DNA is then nicked and it is cleaved into smaller fragments by membrane-bound endonucleases, allowing a single strand to enter the cell through the membrane-spanning DNA translocation channels. Okay, so as a double-stranded DNA is bound to the cell surface receptor, what happens? It is cleaved into smaller fragments by endonucleases, and this allows a single strand DNA to enter the cell through membrane spanning DNA translocation channels. The transformed DNA then gets integrated into chromosome and replaces the chromosomal DNA fragment by recombination. The integration, however, requires significant nucleotide sequence homology between donating DNA fragment and fragment in the chromosome. Then the easily DNA fragment can be replaced by recombination. In the case of plasmid, the plasmid with donor DNA is inserted during the heat shock or electroporation, and then the cells with the plasmids can be detected by growing these cells in a growth media that is supplemented with a specific antibiotic or say any other selectable marker. Okay, so this is how transformation is done, or this is the concept of transformation. So I hope you have a clear concept. Now you had clarified your concept. So this is a simple diagrammatic representation where you can see a plasmid is ligated, DNA is ligated to a plasmid, and then through heat shock treatment it is transformed. Some of your bacterial cells will get incorporated those plasmids, and then if your plasmid has a good selectable marker, you can check those by plating on a antibiotic resistant. For antibiotic resistance, you can check your plasmid that is transformed by using that particular antibiotic in your culture media. Okay, this method also has different applications. So transformation is thus a widely adopted method for altering DNA and incorporating it into another bacterial host cell where it can be expressed. Okay, so thank you for watching. Do like my videos, do share my videos, and do subscribe to my channel. Thank you.